Hello everyone, welcome to today's lecture on the fascinating topic of bioenergetics of exercise and training. Understanding how our bodies transfer and utilize energy is crucial for designing effective and efficient training programs. By grasping how energy is made available for different types of exercise and how we can modify energy transfer through specific training regimens, we can optimize performance and achieve our fitness goals more effectively. So let's begin by clarifying some essential terminology in bioenergetics, which is the study of the flow of energy within biological systems. Bioenergetics primarily concerns the conversion of macronutrients, carbohydrates, proteins, and fats into usable forms of energy. These macronutrients contain chemical energy stored within their bonds, and it's the breakdown of these bonds that provides the energy necessary for biological work, including muscle contraction during exercise. The breakdown of large molecules into smaller ones associated with the release of energy is termed catabolism. For example, when proteins are broken down into amino acids, that's a catabolic process. On the other hand, the synthesis of larger molecules from smaller ones requires energy and is known as anabolism. Building muscle proteins from amino acids after a workout is an anabolic process. Reactions that release energy are called exergonic reactions and are generally catabolic. In contrast, reactions that require an input of energy are known as endergonic reactions. These include anabolic processes and the contraction of muscle fibers. When we talk about metabolism, we're referring to the total of all the catabolic, exergonic, and anabolic, endergonic reactions in a biological system. Essentially, it's the sum of all the chemical processes that occur within a living organism to maintain life. A crucial molecule in this energy exchange is adenosine triphosphate, ATP. ATP acts as an intermediate that allows energy transfer from exergonic to endergonic reactions. Think of ATP as the energy currency of the cell, and it stores and transports chemical energy within cells. Without an adequate supply of ATP, muscular activity and growth wouldn't be possible. If we look deeper into the structure and function of ATP, ATP is composed of an adenosine molecule bonded to three phosphate groups. Adenosine itself consists of adenine, a nitrogen-containing base, and ribose, a 5-carbon sugar. The three phosphate groups are connected by high energy bonds, and it's the breaking of these bonds that releases energy for cellular processes. The breakdown of one molecule of ATP to yield energy is known as hydrolysis because it requires one molecule of water. This reaction is catalyzed by an enzyme called adenosine triphosphatase, ATPase. There are specific types of ATPase enzymes that play different roles. Myosin ATPase catalyzes ATP hydrolysis for muscle contraction. Calcium ATPase helps pump calcium into the sarcoplasmic reticulum, aiding muscle relaxation. Sodium potassium ATPase maintains the concentration gradients of sodium and potassium ions across the cell membrane after depolarization, which is essential for nerve impulse transmission. The hydrolysis reaction can be represented in formula where ADP stands for adenosine diphosphate, ATP -1 phosphate group, Pi is inorganic phosphate, and H plus is a hydrogen ion proton. Further hydrolysis of ADP can yield adenosine monophosphate, AMP. ATP is classified as a high-energy molecule because it stores significant amounts of energy in its phosphate bonds. However, muscle cells store only limited amounts of ATP. At any given time, the body stores approximately 80 to 100 grams of ATP, which isn't enough for prolonged activity. Therefore, ATP must be continually resynthesized to meet the energy demands of exercise. To replenish ATP, our bodies rely on three basic energy systems: phosphagen system, glycolysis, oxidative system. These systems work together to ensure a continuous supply of ATP during different types of physical activity. Importantly, all three systems are active at any given time, but the extent to which each contributes depends primarily on the intensity of the activity and secondarily on its duration. In discussions about bioenergetics, you'll often hear the terms anaerobic and aerobic metabolism. Anaerobic processes don't require oxygen and include the phosphagen system and glycolysis, which occur in the sarcoplasm of muscle cells. Aerobic mechanisms depend on oxygen and involve the oxidative system, which takes place in the mitochondria of muscle cells. An important point to note is that of the three macronutrients, carbohydrates, proteins, and fats, only carbohydrates can be metabolized for energy without the direct involvement of oxygen. This makes carbohydrates crucial during high-intensity anaerobic activities. The phosphagen system provides ATP primarily for short-term, high-intensity activities such as sprinting, heavy weightlifting, and explosive movements like jumping. It's highly active at the start of all exercise regardless of intensity. This system relies on two key reactions: the hydrolysis of ATP itself, the breakdown of creatine phosphate, CP, also known as phosphocreatine, PCR. The enzyme creatine kinase catalyzes the reaction between ADP and CP to form ATP and creatine. This reaction is important because it rapidly replenishes ATP by transferring a phosphate group from CP to ADP. CP stores are limited, but they are essential for providing immediate energy during short, intense bursts of activity. Another important reaction is the adenylate kinase reaction, also known as the myokinase reaction. And this reaction is significant because it not only helps regenerate ATP but also produces AMP, which is a powerful stimulant of glycolysis, preparing the body to produce more ATP through other means. The phosphagen system operates under the law of mass action, which states that the direction and rate of a chemical reaction are driven by the concentrations of the reactants and products. When ATP levels decrease during intense exercise, ADP and Pi levels rise, which stimulates creatine kinase and adenylate kinase reactions to produce more ATP. Once ATP levels are restored, these reactions slow down or reverse. These reactions are considered near equilibrium reactions because they can quickly adjust to the body's energy demands. This system is especially important during the initial stages of exercise and during activities that require quick bursts of energy. Although the phosphagen system is highly effective for immediate energy supply, the stores of ATP and CP in the muscles are limited. ATP stores cannot be completely depleted because it's essential for basic cellular functions. Research indicates that ATP concentrations may decrease by up to 50-60% during intense exercise, leading to muscle fatigue. Interestingly, type 2 fast twitch muscle fibers contain higher concentrations of creatine phosphate than type 1 slow twitch fibers. This means individuals with a higher proportion of type 2 fibers may be better at quickly replenishing ATP during high intensity and aerobic activities. Moving on to the second energy system, glycolysis is the breakdown of carbohydrates, either glycogen stored in the muscles or glucose delivered in the blood, to resynthesize ATP. Glycolysis involves multiple enzymatic reactions in the sarcoplasm and produces ATP at a slower rate than the phosphagen system, but has a higher capacity due to a greater supply of glycogen and glucose. The end product of glycolysis is pyruvate, which can follow one of two pathways. When oxygen is limited and energy demand is high, such as during intense exercise, pyruvate is converted to lactate. This process is catalyzed by the enzyme lactate dehydrogenase. It's often referred to as anaerobic glycolysis or fast glycolysis. ATP resynthesis occurs at a faster rate but is limited in duration because of the accumulation of hydrogen ions, which can lead to a decrease in pH, metabolic acidosis. When oxygen is sufficient and energy demand is lower, pyruvate is transported into the mitochondria. Here, it's converted into acetyl-CoA and enters the Krebs cycle for further ATP production. This pathway is known as aerobic glycolysis or slow glycolysis. ATP resynthesis is slower but can continue for longer periods, supporting sustained activities like long-distance running. A common misconception is that lactate causes muscle fatigue during intense exercise. However, lactate itself is not the culprit. The real issue is the accumulation of hydrogen ions resulting from ATP hydrolysis, which leads to a decrease in intracellular pH, inhibiting muscle contraction and enzyme activity. In fact, lactate can be used as an energy substrate, especially in type 1 muscle fibers and the heart. It can also be transported to the liver, where it's converted back into glucose via the Cori cycle, contributing to the body's energy supply during recovery. The production of lactate increases with exercise intensity and depends on muscle fiber type. Type 2 fibers produce lactate at a higher rate than type 1 fibers due to their higher glycolytic enzyme content. Blood lactate concentrations reflect the balance between lactate production and clearance. During high-intensity exercise, lactate accumulates because production exceeds clearance. However, lactate levels typically return to resting levels within an hour after exercise, depending on factors like exercise duration, intensity, training status, and recovery methods. Active recovery, light exercise post-activity, can enhance lactate clearance compared to passive recovery. Trained individuals also tend to clear lactate more efficiently than untrained individuals. The overall reaction for glycolysis when pyruvate is converted to lactate can be summarized in formula, and this reaction yields a net gain of 2 ATP molecules per glucose molecule, providing energy for short to moderate duration activities. Remember, only carbohydrates can be metabolized without oxygen, making them essential during anaerobic metabolism. Therefore, adequate carbohydrate intake is crucial for athletes engaged in high-intensity training. During intense exercise, the accumulation of hydrogen ions leads to metabolic acidosis, which can contribute to fatigue by inhibiting muscle contraction and enzyme activity. This underscores the importance of training programs that enhance the body's ability to buffer and clear these ions, improving performance and delaying fatigue. Understanding these energy systems has practical implications for designing training programs. High intensity, short duration training emphasizes the phosphagen system. Activities include sprints, heavy lifting, and plyometrics. Training can increase creatine phosphate stores and enhance enzyme activity for rapid ATP resynthesis. Moderate intensity, moderate duration training relies on glycolysis. And activities include 400 to 800 motor runs or circuit training. Training can improve glycolytic capacity and lactate threshold, allowing athletes to perform at higher intensities before fatigue. Low intensity, long duration training depends on the oxidative system, not covered in detail here. Activities include long distance running or cycling. Training enhances mitochondrial density and aerobic enzyme activity, improving endurance. In summary, the body's energy systems work together to meet the demands of different types of exercise. By understanding how these systems function and interact, we can tailor training programs to optimize energy production, delay fatigue, and enhance overall performance. Whether you're an athlete aiming for peak performance or a coach designing training regimens, knowledge of bioenergetics is a powerful tool. It allows for the creation of specific training protocols that target the appropriate energy systems, leading to more efficient and effective results.